So, hello, my friends. Today we are discussing the advancement in the bioinformatics and genomics and biomaterials. Okay, so now can take the questions. Which of the following is a significant advancement in bioinformatics for genome sequencing? Genome sequencing के लिए कौन सा अभी एडवांसमेंट हुआ है बायोइन्फॉर्मेटिक्स के फील्ड में? तो ये होता है नेक्स्ट जनरेशन सीक्वेंसिंग। नेक्स्ट जनरेशन सीक्वेंसिंग क्या होता है? ये एक हाई थ्रूपुट टेक्नोलॉजी है दैट हैज ये रिवॉल्यूशनाइज्ड जीनोम सीक्वेंसिंग कर दिया है अनलाइक ट्रेडिशनल सैंगर सीक्वेंसिंग एंड नेक्स्ट जनरेशन सीक्वेंसिंग में क्या होता है अलाउ द साइमल्टेनियस सीक्वेंसिंग फॉर मिलियंस ऑफ डीएनए फ्रैगमेंट मेकिंग इट फास्टर चीपर एंड मोर एफिशिएंट इन जीनोमिक रिसर्च मोर एफिशिएंट इन जीनोमिक रिसर्च में विच कंप्यूटेशनल टूल कौन सा कंप्यूटेशनल टूल जो है कॉमनली यूज्ड हो रहा है फॉर द स्ट्रक्चरल बायोइन्फॉर्मेटिक्स टू प्रिडिक्ट प्रोटीन स्ट्रक्चर प्रिडिक्ट प्रोटीन के स्ट्रक्चर को प्रिडिक्ट करने के लिए कौन सा अभी टूल जो है वो यूज हो रहा है? ये होता है पाइमोल। पाइमोल जो है क्या है? पावरफुल मॉलिक्यूलर विजुअलाइजेशन टूल है। टूल वाइडली यूज हो रहा है फॉर स्ट्रक्चरल बायोइन्फॉर्मेटिक्स। इट इज प्राइमरी यूज्ड to visualize protein structure, nucleotides structure, and molecular dynamic molecular dynamic stimulus. What is the primary function? क्या होता है जीनोम वाइड एसोसिएशन स्टडी का प्राइमरी फंक्शन क्या है? टू अंडरस्टैंड द रिलेशनशिप बिटवीन जीन्स एंड फिनोटाइप। जीन्स एंड फिनोटाइप्स के बीच में जो है रिलेशनशिप को बताता है ये। जीवास जो है जीनोम जीनोम वाइड एसोसिएशन स्टडी जो है मेथड है। ये एक मेथड है यूज टू आइडेंटिफाई जेनेटिक वेरिएंट एसोसिएटेड विथ डिजीज और ट्रेट्स इन द पॉपुलेशन। इट हेल्प्स इन एसोसिएटिंग स्पेसिफिक जेनेटिक वेरिएशन विथ डिजीज thereby adding the understanding of प्री प्रीडिपोजिशन जेनेटिक प्रीडिपोजिशन पे ये है। ठीक है। व्हाट इज अ टेक्नोलॉजी व्हाट इज अ रिसेंट टेक्नोलॉजी एडवांसमेंट इन जेनो जेनो जेनोसिक्स फॉर लार्ज स्केल जीनोम सीक्वेंसिंग मोर अफोर्डेबल सो इसका नैनोपोर सीक्वेंसिंग क्या होता है? लाइक the technology development of by Oxford Nanopore technology has been made large-scale genome sequencing more affordable and portable. It uses the nanopore to detect electrical signal generated by nucleosides passing through enables the real-time sequencing with minimal sampling preparation with minimal sample preparations. Which of the following जो है is a goal of functional genomics? फंक्शनल जीनोमिक का फॉलोइंग गोल्स में से कौन सा है? तो ये अंडरस्टैंडिंग द फंक्शन ऑफ जीन एंड देयर रेगुलेशन एलिमेंट द रेगुलेटरी एलिमेंट्स understanding the function of genes and their regulatory elements. Functional genomics is focused on the understanding of how genes and their regulatory elements control cellular function. It aims to identify gene function, expression pattern interaction, often using technologies like RNA sequence. What is the major recent challenge in the field of genomics? Genomics के फील्ड में रिसेंट में कौन सा चैलेंज हुआ है? तो ये मैनेजिंग एंड एनालाइजिंग द वास्ट अमाउंट ऑफ डाटा जनरेटर। मैनेजिंग एंड एनालाइजिंग जो है वास्ट अमाउंट ऑफ डाटा जो अभी जनरेट हो रहा है फॉर उसको एनालाइज करने में। विच वन ऑफ द सिग्निफिकेंट चैलेंज इन जीनोमिक्स is a big data problem. It is in the rapid advancement in the sequencing technology generate massive amount of data that need to be stored, processed, or analyzed. Developing efficient computational method and storage system remains a key challenge. Which of the following is a major recent development in the field of biomaterial? बायोमटेरियल के फील्ड में क्या recent development हुआ है? Synthetic polymers for drug delivery. The development of biomaterial have focused on using synthetic polymer for drug delivery. Delivery system. The polymers can be engineered to release drug at controlled rate, improving the efficacy and reducing the side effect of the treatment. सिंथेटिक पॉलीमर फॉर ड्रग डिलीवरी, विच बायोमटेरियल जो है कॉमनली यूज हो रहा है बोन टिश्यू इंजीनियरिंग के लिए। बोन टिश्यू इंजीनियरिंग के लिए ये हो रहा है हाइड्रो एक्सपेक्टाइट। हाइड्रो एक्सपेक्टाइट जो है नेचुरली हाइड्रो हाइड्रोक्सीटाइट इज नेचुरली ऑकरिंग मिनरल्स formed by the calcium apatite and is widely used in bone tissue engineering. Widely used in bone tissue engineering. It is biocompatible and similar to the similar content of human bones, making it ideal material for bone regeneration. बोन रीजनरेशन के लिए एक आइडियल मटेरियल है, मतलब कैल्शियम बेस्ड मटेरियल है, यूज हो रहा है। ठीक है। Which property of biomaterial is most crucial for the use of medical implant? मेडिकल इम्प्लांट के लिए कौन सा बायोमेडिकल प्रॉपर्टी जो है मोस्ट क्रूशियल है? ये है हमारा बायोकम्पेटेबिलिटी। बायोकम्पेटेबिलिटी है कि नहीं है, इज द मोस्ट क्रूशियल प्रॉपर्टी ऑफ फॉर मेडिकल इम्प्लांट बिकॉज ये जो है इंश्योर करता है मटेरियल does not cause adverse immune response toxicity when placed inside the human body. Biocompatible material are safe and integrated well with the tissue. What is the what is the significance of the single cell RNA sequencing in single cell sequencing RNA sequencing? ये जो है at allow for the study of gene expression at a single cell level पे ये करता है। single cell RNA sequencing is a technique है used for profile gene expression in individual cell, revealing cellular heterogeneity and can't be detected using bulk RNA sequencing method. This method technology is advanced our understanding of cellular function in complex tissue. Which of the following जो है वो key characteristics है of the BioLink used in the 3D bioprinting? 3D bioprinting तो ये है ability to promote टिश्यू ग्रोथ। एबिलिटी टू प्रमोट टिश्यू ग्रोथ। बायोलिंक्स जो है यूज हो रहा है इन 3D बायोप्रिंटिंग are designed to mimic the properties of natural tissue and promote the tissue growth. These materials are usually made from biocompatible substance like hydrogels, which supports the cell attached and proliferations. What does the BLAST algorithm do in bioinformatics? It aligns the sequence and identifies similarities. The BLAST क्या है? BLAST का प्रोग्राम बेसिक लोकल अलाइनमेंट सर्च टूल। बेसिक लोकल अलाइनमेंट सर्च टूल is widely used algorithm for comparing querying sequence against the database of sequence. It helps in identifying regions of similarity which may provide insight into the function of gene or protein based on the evolutionary relationship. Which bioinformatics tool is commonly used to identify regulatory element in DNA sequencing? DNA sequencing ये क्या है? ये आपका ये है genome browser। genome browser क्या आता है? Such as UCSC genome browser is a popular tool है for visualizing genome. It allows user to track genome data, including regulatory element like promoters, enhancing and transition factor binding sites. Which of the following is the recent advancement in bioinformatics for data storage के फील्ड में एंड उसका मैनेजमेंट के फील्ड में क्या हुआ? क्लाउड कंप्यूटिंग फॉर जीनोम एनालिसिस। क्लाउड कंप्यूटिंग क्या होता है? एट सच एज गूगल क्लाउड और अमेजॉन वेब सर्विसेज हैव बीन बिकमिंग द इंक्रीजिंग important in bioinformatics for storing and analyzing vast amount of genomics data. These platforms allow easy scaling and data sharing, making large scale project more feasible. हम लोग क्लाउड पे जो शेयर करते हैं डाटा वो उसके लिए है। Which bioinformatics approach is most commonly used for studying evolutionary relationship between the species? ये क्या है? फाइलोजेनिक जेनेटिक एनालिसिस। फाइलोजेनेटिक एनालिसिस जो है यूज टू कंस्ट्रक्ट Voluntary trees-based genetic similarities for and differences. The approach helps trace the evolutionary history, species, genes, and proteins. CRISPR Cas9 जो है primarily used in gene editing में ये use होता है CRISPR9 Cas9 ये जो revolutionary gene editing tool है it allows precise modification to DNA of living organisms is greatly accelerated genomic research and enabling the study of genome function, providing potential therapeutic for genetic disease. Which of the following is the role of edip- epigenetic in genomic? Studying gene expression change not caused by DNA sequence change. Studying gene का expression कैसे change हो रहा है not causing by DNA sequence change epig epigenetics involves studying how gene expressions जो है is regulated by chemical modification to DNA and histogenes without altering the underlying DNA sequence. This can influence traits, disease, and inheritance pattern. Which of the following is a major goal of medicine? Is that developing treatment based on patients' specific genetic profile? Precision medicines uses genetic information and biomarkers to tailor treatment to individual patient and understanding genetic differences. Doctors and can select the most effective therapies with fewer side effects. Which is the application of functional genomics in cancer research? Identifying cancer genomics in cancer research. What is the application of the functional genomics in cancer research? Identifying cancer causing mutation इसमें जो है तुम्हारा सब हो जाएगा आइडेंटिफाई इसमें क्या करते हैं आइडेंटिफाइंग कैंसर कॉजिंग म्यूटेशन कौन कौन सा म्यूटेशन हो रहा है sequencing the entire genome of tumor cell understanding how gene expression influence the tumor cell behavior all of the functional genome in a cancer research involving identifying mutation sequencing tumor genome and analyzing the gene expression pattern to better understanding how specific gene contribution to the cancer development and progression. Which of the following is an example of natural polymer used in biomaterial for wound healing? वाउंड हीलिंग के केस में कौन सा एग्जांपल है नेचुरल पॉलीमर का तो हम लोग कोलेजन यूज करते हैं कोलेजन collagen is a naturally occurring protein found in the connective tissue. It is commonly used in biomaterial in for wound healing. घाव को भरने के लिए यूज होता है it is a biocompatible ability to promote cell growth and facilitate tissue regeneration. Which type of biomaterial जो है commonly used in cardiovascular strength? तो ये होता है स्टेनलेस स्टील जो है पार्टिकुलरली मेडिकल ग्रेड स्टेनलेस स्टील इज कॉमनली यूज फॉर कार्डियोवास्कुलर स्टेंट यूज फॉर स्ट्रेंथ फ्लेक्सिबल एंड बायोकम्पैटिबिलिटी it ensures the strength integrity of the stent while being safe for implementation in human body. What is the purpose of using biocompatible hydrogel in tissue engineering? टिश्यू इंजीनियरिंग में क्या है तो ये एक्ट करता है एक्ट एज ए स्ट्रक्चरल सपोर्ट स्कैलफोल्ड बायोकम्पेटेबल हाइड्रोजेल जो है वाइडली यूज्ड एज ए स्कैलफोल्ड इन टिश्यू इंजीनियरिंग बिकॉज इट कैन mimic the extracellular matrix, support cell growth, and facilitate nutrient diffusion while being able to absorb and retain water. Which of the following is a challenge? कौन सा challenge है when using 3D bioprinters, regenerative medicines? जो है lack of suitable biolinks. One of the main challenges in 3D bioprinting is the development of suitable biolinks that support cell viability, promote tissue formation, and then the print at high resolution. Biolinks need to closely mimic the mechanical and biological properties of the target issue. What is the primary advantage किया है for long read sequencing technologies such as PacBios and Oxford Nanopods over a short read sequencing. They are less expensive, they are faster, they provide more accurate sequencing result. are better for sequencing complex genome. इसका क्या आंसर होगा? इसका आंसर होगा डी। For it is for better sequencing complex genomic region के लिए हम लोग यूज करते हैं, तो प्राइमरी एडवांटेज यही है sequencing टेक्नोलॉजी का। Long read sequencing technologies such as those from the PacBio and Oxford Nanopods provide a longer sequencing read and particularly useful for sequencing complex organic genome like repetitive sequencing and structural variations variant are challenging for the short read technologies. What is the primary challenge associated with इस Cas9 Cas gene therapy? इसके लिए क्या है? Potential target effect of target effects. The main challenges of the Cas9 in gene therapy is the potential for the off target effect. Those Cas9 enzymes could cut unintended part of genome, leading to the unwanted genetic change. Which of the following is a key feature? कौन सा इसमें जो है की फीचर है of scaffold tissue engineering के लिए? तो ये they should degrade over time to allow tissue formation. Scaffold tissue in engineering is designed to degrade over time, allowing the newly formed tissue to take over the function of scaffold. This gradual degradation and support the tissue regeneration while minimizing the need for long term implant. Which of the following जो है are biocompatible material is commonly used for bone regeneration के लिए calcium phosphate based ceramics like hydroxypeptide are often used in bone regeneration due to the similarities of natural bone materials minerals, and they promote क्या है ये Ops conductivity, supporting bone growth and integrating with surrounding tissue. What is the main function of reference genome in bioinformatics? May to serve as a standard template for comparing genetic variation. A reference genome is a digital representation of genetic material and a species, serving as a standard template. It allows researchers to compare and identify variations, mutations, and structural difference between the individual or populations. Main reference genome. Which tool is used? Which tool is used to identify an elementary relationship between the gene and from the different species? Custal Omega. Omega is a multiple sequence alignment tool used for aligning gene or protein from the different species. They helps to identify voluntary relationship, aligning sequence, and determining the conserved region across species. Which bioinformatics technique is used to predict protein-protein interaction? इसका होगा स्ट्रिंग डाटाबेस। स्ट्रिंग जो है डाटाबेस वाइडली यूज होता है बायोइन्फॉर्मेटिक्स टूल्स टू प्रिडिक्ट एंड एनालाइज द protein-protein interaction based on known or predicted relationship, including physical interaction, co-expressions, and functional association. What is the primary use of Casper interference? Casper interference to inhabitate gene expression. So Cas9 method is used for catalytically dead Cas9 to bind to the gene to promote a region, preventing transcriptions, and used to inhibit gene expression without altering the gene sequence. Which of the following genome technique? Which of the concept following genome technique is the most useful for identifying structural variant such as deletion and duplication in genomes? इसका होगा जी क्रोमोसोमल माइक्रो एरे एनालिसिस। क्रोमोसोमल मैक्रो एरे एनालिसिस जो है इज हाईली इफेक्टिव फॉर डिलीटिंग structural variant like deletion, duplication, copy number variations in genome. It allows high resolution detection for these variant. What is the role of methylation methylation and in the regulation of gene expression के लिए? It inhibited gene transcription by adding methyl group to DNA. मिथाइल ग्रुप को डीएनए में डालता है, तो डीएनए मिथाइलेशन typically silence gene expression by adding methyl groups to cytosine residue in DNA, particularly to promote regions that inhibited transcription factors binding and gene expression, which sequencing technology is commonly used for high throughout RNA sequencing. ये क्या है? इल्यूमिना sequencing is most widely used platform for high throughput RNA sequencing. RNA sequencing offers accurate and efficient sequencing of RNA samples to study the gene expression. फिर आता है तुम्हारा बायोमटेरियल इज मोस्ट कॉमनली यूज्ड इन फैब्रिकेशन ऑफ हाइड्रोजेल्स दैट वाउंड हीलिंग एप्लीकेशन वाउंड पॉलीइथिलीन ग्लाइकॉल पेग पेग का यूज करते हैं हम लोग वाउंड हीलिंग के लिए। ये क्या होता है? फ्रीक्वेंटली यूज इन द डेवलपमेंट ऑफ हाइड्रोजेल्स वाउंड हीलिंग ड्यू टू द biocompatibility, flexibility, and ability to retain water, which support tissue hydration and cell migrations. Which of the following biomaterial is used for the development of bio क्या लिखा है ये? जो भी सर्जिकल सरफेस के लिए, तो ये आपका सिंथेटिक बायोडिग्रेडेबल पॉलीमर्स, सिंथेटिक बायोडिग्रेडेबल पॉलीमर्स such as a polyglycolic acid and polyacetic acid are commonly used for resorptionable surgical stretcher. क्या है ये? पता नहीं। These generally dissolve in the body, eliminating the need of removal after tissue healing. What is the primary benefit of using nanomaterials in drug delivery system? तो ये already हम लोग देख चुके हैं. It allows the control and target drug release. Nanomaterials used in drug delivery system because it can engineer to carry drug to a specific site in the body, releasing the drug in the controlled manner, improving the efficacy of the treatment while minimizing the side effect. Which of the following is commonly used biocompatible material for dental implants? For dental implants के लिए क्या होता है? A टाइटेनियम का यूज करता है डेंटल इम्प्लांट्स के लिए, दांत के लिए. डेंटल इम्प्लांट्स फॉर एक्सलेंट बायोकम्पेटेबल स्ट्रेंथ एबिलिटी टू इंटीग्रेट विथ बोन, ए प्रोसेस कॉल्ड, ये जो भी प्रोसेस है. which of the which of the following method is used to sequence ancient DNA from archaeological sample के लिए तो next generation sequencing क्या होता है? revolutionize study ancient DNA by enabling high throughput sequencing of fragmentation and degradation of DNA from the archaeological sample, allowing researcher to construct a genome of extinct species. What is the major advantage of using single cell sequencing in genome? ये already हम लोग देख चुके हैं. It enables the gene expression of the cell individual. ये क्या होता है? Researchers to analyze the gene expression profile of individual cell, providing insight into cellular heterogeneity within the tissue and uncovering the new layer of gene regulations. What is the significance of long-read sequencing technologies such as PacBios, Oxford Nanopores, and genome assembly? इसका होता है they can assemble complex and repetitive regions of the genome more effectively. Long read sequencing technologies such as BioPax and Oxford Nanopores is a crucial for assembling complex and repetitive genomic regions which are difficult to resolve. The short read sequencing, these technologies improve genome assembly, particularly for the genome with a repetitive sequence. Which type of biomaterial जो है being developed for the use of 3D bioprinting of tissues? ये होता है हाइड्रोजेल्स. हाइड्रोजेल्स हम लोग पहले देख चुके हैं. Biomaterials is most commonly used in the bone implant and implant for the ability to promote bone growth. ये हम लोग हाइड्रोक्सपेटिक का यूज करते हैं। ये कैल्शियम फॉस्फेट कंपाउंड है, केमिकली सिमिलर टू द ह्यूमन बोन्स मिनरल होता है एंड ये बायोकम्पेटेबल होता है एंड ऑस्ट्रो conductive properties make it ideal for promoting bone growth in the bone implants. What is the major challenge for using stem cell in tissue engineering? Stem cell का controlling stem cells differentiating and behavior. One of the key challenges क्या है in tissue engineering? In tissue engineering is controlling stem cell differentiation behavior. Stem cells have potential to become various cells types, but directing their differentiation into the specific tissues and controlling the reproducible manner is still major obstacles. Which of the following is a tool is primarily used to identify conserved gene families across the multiple species? तो ये था ऑर्थोबीटी। ऑर्थोबीटी इसे क्या है, डाटाबेस है जो हेल्प करता है आइडेंटिफाई ऑर्थोलॉगस लॉगस जीन फैमिलीज gene families across the multiple species. Orthologs are the gene in different species that evolved from the common ancestral gene, and identifying them helps to in understanding the evolutionary relationship. ये क्या होता है, बायोइन्फॉर्मेटिक्स। इन बायोइन्फॉर्मेटिक्स व्हाट इज द प्राइमरी पर्पस ऑफ जीन ऑर्थोलॉजी ऑन्टोलॉजी ऑन्टोलॉजी जीओ ओनोमस? ये क्या होता है, क्लेरिफाई जीन फंक्शनल इनटू बायोलॉजिकल प्रोसेस, मॉलिक्यूलर फंक्शन एंड सेलुलर कंपोनेंट। जीन ऑन्टोलॉजी क्या होता है? जीन ऑन्टोलॉजी होता है तुम्हारा प्रोवाइड करता है स्टैंडर्डाइज वे टू डिस्क्राइब जीन फंक्शन अक्रॉस स्पीसीज। These annotation categorize gene product based on their biological process, molecular functions, and cell component, helping researchers understanding the gene role. What is the function? What is which of the following tool is best suited for detecting genomic variant like SNPs and indels? कहता है गटक। जीनोम एनालिटिक टूल किट जो है, जिसको घटक बोलते हैं, इज वन ऑफ द मोस्ट कॉमनली यूज्ड टूल फॉर वेरिएंट कॉलिंग में यूज था, इंक्लूडिंग डिटेक्शन ऑफ सिंगल न्यूक्लियोटाइड्स, पॉलीमॉर्फिज्म एंड सिंगल न्यूक्लियोटाइड्स पॉलीमॉर्फिज्म एंड इंसर्शन और डिलीशन इंडेल्स फ्रॉम द सीक्वेंसिंग डाटा। Which is the key benefit using RNA sequencing over macro arrays. The RNA sequencing detect both known and unknown gene. RNA sequencing जो है डिटेक्ट करता है तुम्हारा both known और unknown जीन को। is capable of detecting all expression gene including novel gene isoforms, whereas microarray are limited to detecting only known gene for which probes exist. Which of the following which provide more comprehensive analysis for gene expression? Which is the purpose using whole genome sequencing in personalized medicine? तो इसका भी हो जाए आइडेंटिफाइंग जेनेटिक म्यूटेशन एंड प्रिडिक्ट द डिजीज रिस्क। फिजिक्स होगा इसमें, डिजीज रिस्क को अपने को नहीं बचा इसमें। पेटी ज़्यादा वो हो गया है, हैंग हो गया। होल जीनोम whole genome sequencing जो है helps to identify genetic mutation across the entire genome, which can provide insight into disease risk, predispositions, predispositions to conditions and response to the treatment. Then this forms the foundation of personalized medicine, where treatments are tailored based on genetic profiles. Which of the following method to analyze gene expressions across different tissues in which human body in which human body which of the following method to analyze the gene expression across the different tissues in human body? इसमें होता है आरएनए सीक्वेंसिंग में क्या आता है हाई थ्रूपुट सीक्वेंसिंग टेक्निक is used to analyze gene expression level across the different tissue. It allows researchers to quantify the transcription, identifying gene are active in the various tissue types. What is the purpose of utilizing Cas9 genome research? ये हम लोग जनरली यूज करते हैं genome silencing. Identify the function of genome silencing. Cas9 used to genome researcher to create target gene knockout by silencing specific gene. Researchers can study the function and the role in the disease development and other biological process. अच्छा, ज़्यादा नहीं पूछा. What is the major limitation क्या है using a single nucleotides polymorphism में for disease association studies के लिए क्या है they do not account for environmental factor, they do not account for are using genetic studies but they can't account for genetic environmental factors that influences the disease susceptibility. Environmental factors such as diet, lifestyle and other often interact with genetic variant of contribution of disease, making SNPs SNPs क्या हुआ तुम्हारा सिंगल न्यूक्लियोटाइड polymorphism alone insufficient in predicting the complex disease. Which biomaterial कौन सा biomaterial है is primarily used for creating scaffolds in bone tissue engineering. bone tissue के लिए हम लोग ये करते हैं हाइड्रोक्सीपेटाइट. Which of the following is the advantage of using 3D printing in tissue engineering? इसके लिए क्या है ability to create complex tissue structure within high precision. And the key advantage using biocompatible hydrogels for drug delivery system के लिए क्या यूज करते हैं ये सिस्टम करते हैं they can release drug in controlled and sustainable manner. Biocompatible hydrogels जो है ideal for drug delivery because they encapsulate drug and release them into a controlled sustainable manner, improving drug efficacy and minimizing the side effect. What is the most common method for sterilizing the biomaterials using medical implant? होता है इसमें होता है गामा रेडिएशन से हम लोग सारे स्टेरिलाइजेशन कर देते हैं. Gamma radiation is commonly used to sterilize medical implants and other biomaterials due to the ability to kill bacteria and other pathogens without damaging the material. In particularly effective for sterilizing the material that cannot withstand heat. What is the significance of using high throughput sequencing technique in metagenosis? तो इसमें ये होता है it allows for the identification of individual species in complex microbial communities. It allows the identification of individual species in a complex microbial communities. Explanation क्या है इसका high throughput sequencing metagonics capable of studying of complex microbial communities by sequencing DNA directly from the environment sample. This allows the identification of species those can't cultured in laboratory, जो cultured नहीं हो पाता laboratory में. What is the significant limitation of using RNA बारकोडिंग इन स्पीसीज आइडेंटिफिकेशन, इन स्पीसीज आइडेंटिफिकेशन के लिए है। तो इसका आंसर होगा डी, इट रिक्वायर्स द रेफरेंस डाटाबेस with sequence फ्रॉम मेनी स्पीसीज, विद सीक्वेंस फ्रॉम मेनी स्पीसीज। विच ऑफ द फॉलोइंग मटेरियल जो है, कौन सा मटेरियल जो है कॉमनली यूज्ड हो रहा है इन द डेवलपमेंट ऑफ कॉन्टैक्ट लेंसेज। कॉन्टैक्ट लेंस में सिलिकॉन हाइड्रोजेल्स का यूज हो रहा है। सिलिकॉन हाइड्रोजेल जो होता है डेवलपमेंट ऑफ कॉन्टैक्ट लेंसेज इज हाई ऑक्सीजन permeability. It is essential for eye health as well as the flexible and comfort for long term wear. Primary benefit of using nanostructured biomaterials in tissue engineering is a, enhanced tissue regeneration through improved cell signaling. Nanostructured biomaterials जो है designed to mimic the extracellular matrix at a nanoscale, which promote करता है better cell adhesion, pyro pyro proliferations and differentiation, leading to enhanced tissue regeneration. Which of the following bio informatics tool is primarily used for protein structure prediction? प्रिडिक्शन के लिए क्या सा यूज होता है? अल्फा फोल्ड डेवलप्ड बाय डीप डीप माइंड एडवांस मशीन लर्निंग सिस्टम डिजाइन टू प्रिडिक्ट प्रोटीन स्ट्रक्चर फ्रॉम अमीनो acid sequence. It has revolutionized the field of structural bioinformatics by accurately predicting the 3D structure of protein. What is the purpose of sequence alignment in bioinformatics? तो ये होता है टू फाइंड द कंजर्व जेनेटिक रीजन अक्रॉस द डिफरेंट ऑर्गेनिज्म। सीक्वेंस अलाइनमेंट क्या होता है? इट इज द प्रोसेस ऑफ arranging sequence of DNA, RNA, or protein to identify regions of similarity. It helps to identify conserved genetic regions across different species, which can provide insight into their evolutionary history and functional importance. विच ऑफ द फॉलोइंग जो है यूज होता है टू रिप्रेजेंट द लार्ज स्केल जीनोमिक्स डाटा विजुअली इन बायोइन्फॉर्मेटिक्स में? तो जीनोम ब्राउजर का हम लोग यूज करते हैं। जीनोम ब्राउजर जो है सच एज UCSC जीनोम ब्राउजर और इनसेल used to visualize large scale genomic data, including genome annotations, variant call, expression level. When these browsers provide an interactive interface to explore the genomics data. Which of the following जो है एक major challenge when analyzing the big data in bioinformatics? तो computational complexity and data integration जो है तुम्हारा complex हो जाएगा। Which of the following is the main purpose? So which of the following is the main purpose of using whole exome sequencing? And the sequence only the exomes of the gene code for protein. The whole exome sequencing जो है focus करता है sequencing of exonic region of genome, which are the part of gene that code for protein. This approach is the most cost-effective whole genome sequencing, while still providing valuable insight into coding mutations and can lead to diseases. What is the primary benefit? क्या है primary benefit of using single cell RNA sequencing में? क्या है? Yet it provides insight into individual cell heterogeneity. Single cell RNA sequencing जो है allow for the analysis of gene expression at a level of individual cell. It helps to identify cellular diversity within the tissue, revealing how different cell contribution to biological process and disease. Which of the following, which sequencing technology is known for its ability to read long DNA sequence, making it useful or assembling complex genome? तो ये हो जाएगा तुम्हारा बायोपैक सिक्वेंसिंग जो है तुम्हारा लार्ज डीएनए को रीड कर सकता है। ये क्या ऑल्सो नोन एज सिंगल मॉलिक्यूल रियल टाइम sequencing is known for its ability to generate a long read, making it ideal for assembling complex genome, especially for those with repetitive sequence such as plant and mammalian genome. Which of the following tool is commonly used to detect mutation in cancer genomes? तो ये होता है डी कॉस्मिक कैटलॉग ऑफ स्टोमेटिक म्यूटेशन इन कैंसर। ये क्या है? कॉम्प्रिहेंसिव सोर्स है दैट catalogs somatic mutation found in the cancer genome. It provide information on the frequency and the type of mutation in various cancer type, adding in cancer research and clinical application. Which material is often used for creation of artificial heart valve due to the biocompatibility and durability? तो इसमें हम लोग यूज करते हैं पायरोलेटिक कार्बन जो है वो तुम्हारा आर्टिफिशियल हार्ट वेल बनाने के काम आता है। कॉमनली यूज फॉर क्रिएटिंग आर्टिफिशियल हार्ट वेल ड्यू टू द हाई बायोकम्पेटिबिलिटी ड्यूरेबिलिटी एंड रेजिस्टेंस टू वेयर इट मिमिक्स द मैकेनिकल प्रॉपर्टीज ऑफ नेचुरल हार्ट टिश्यू। Which of the following is a key characteristics of biomaterial used for drug delivery system? तो इसमें आता है दे मस्ट हैव कंट्रोल डिग्रेडेशन रेट। तो होता है बायोमटेरियल्स यूज्ड इन ड्रग डिलीवरी सिस्टम आर टिपिकली डिजाइंड टू डिग्रेड in controlled manner in to release the drug over time. This controlled degradation ensure the sustained and localized release of drug, improving the therapeutic outcomes. Which one of the following biopolymer is commonly used in the fabrication of tissues scaffold regenerating medicine? तो ये है तुम्हारा आपका सी चित्सोन। चित्सोन Chitson is a natural biopolymer derived from the chitin. Is commonly used for fabrications of tissue scaffold due to biocompatibility, biodegradability, and ability to support cell adhesion and growth. Which of the following जो है common method used for fabricate synthetic bone scaffold for tissue engineering? तो ये है तुम्हारा सी 3D बायोप्रिंटिंग। 3D बायोप्रिंटिंग के हेल्प से हम लोग फैब्रिकेटेड सिंथेटिक स्कैलफोल्ड जो है बनाते हैं। इसे कटिंग एज ये क्या है? एक कटिंग एज टेक्निक है यूज फॉर फैब्रिकेटिंग सिंथेटिक बोन स्कैलफोल्ड। इट अलाउज फॉर प्रिसाइज कंट्रोल ओवर द प्लेसमेंट ऑफ द सेल growth factor and biomaterials, enabling the creation of complex tissue structures for bone regeneration. Which is the main advantage of using biomaterials for wound healing application? Wound healing application के they enhance tissue repair by providing a scaffold for cell growth and guiding the healing process. ये सही लग रहा है। Scaffold for the growth cell growth and guiding the healing process. Biomaterial used in wound healing to provide scaffold to support growth and migration of the cell, promote uh, an angiogenesis. It helps in the tissue regeneration. These material play करता है crucial role in accelerating the healing process, improving the quality and healed tissue headed and healed tissues. Which bioinformatics tool is used for functional annotation newly sequenced genome? InterProScan. इंटरप्रोस्कैन क्या होता है? इंटरप्रोस्कैन इज अ बायोइन्फॉर्मेटिक्स टूल दैट जो कि स्कैन कर देता है प्रोटीन सीक्वेंसिंग अगेंस्ट मल्टीपल डाटाबेस टू प्रिडिक्ट functional domains, motifs, and families. It helps in the annotation newly sequenced genome by providing insight into the protein function. What is the main limitation? क्या है? What is the main limitation of whole genome sequencing WGS in clinical setting? क्लिनिकल सेटिंग में क्या है? तो ये है तो हाई कॉस्ट लार्ज डाटा स्टोरेज रिक्वायरमेंट होता है इसमें। होल जीनोम सीक्वेंसिंग में जो है जनरेट करता है लार्ज अमाउंट ऑफ डाटा। इट इज कॉस्टली होता है एंड लिमिट इट्स वाइड स्प्रेड यूजिंग क्लिनिकल सेटिंग। Data storage and computational resource costs are significant challenge in routine clinical implementation of WGS. So last question, what is the role of stem cell in regenerative medicine? Regenerative medicine method they differentiate into specific cell type to repair और regenerate damaged टिश्यू के लिए स्टीम सेल क्या होता है प्लेज ए crucial role in regenerative medicine because they have a ability to differentiate into different cell type. This allows them to use for repairing regenerating damaged tissue such as cast the stem cell therapies for injuries and degenerative disease. So thank you for listening.